RNA can be visible. The probe will not match means the scene. So never can be visible. Never can be visible on our slide. So probe is very very important tool. What is mean by probe? Now it's a labeled. It is a short labeled, and it is which is a targeted sequence actually by using radioactive atom and to find the radioactive finally to find the radioactive atoms. And the technique we are using autoradiography for southern blotting. The probe labeled molecules to find the specific DNA present or not. So we can visible in finally our specific DNA which can be present or not. The gel electrophoresis, and that is our final for the slide. So I will give the introduction for gel electrophoresis. The gel electrophoresis techniques, which is immobilist techniques, and why I'm treating with this important means we are taking a tray, okay, and then we adding some of buffer BSS buffers or buffer, and then we arranging the set arranging the apparatus like gel. We are adding the gel which consists of a particular genome, okay. So we are cutting and taking the genome that is to be incorporated into the gel now is present. All the genes can be present in a gel only as now, and then we add for nitrocellulose membrane is also called as nitrocellulose membrane, okay. In above that, to add 3 sheets of filter paper, and then to add more than 5 sheets of paper tissues, and then to add glass plate, and then to add some of the weight. It may be exceeding 0.75 kg. What is the purpose is actually we are going to do for the filtration, and how our DNA can be filtered and where it can be filtered. So that is our concept. So the DNA can be is present is now for gel, okay. In the buffer has to be raised. We are giving the electric power, and buffer can be raised to reach gel, and the gel of which incorporated into DNA that can be moved towards the membrane. So what is the role of membrane? That nitrocellulose membrane can be incorporated. It is immobilized. It is a lack which lack the DNA. It is or it to be track the DNA. It cannot be moved upstairs from the sheets. Okay, so it is that major role. It has to be tracked the DNA, tracked the RNA. So that is a major role. After the buffer can be raised to membrane, the DNA can be tracked over the membrane. After the buffer can be raised to filter paper, and that is for unbound probe. Okay, so this is only the condition for probe bound, and that is for unbound probe. So we need to discard other things and to take a membrane. Okay, so we take a membrane only. Why? The DNA can be incorporated into the membrane only. So we are taking a DNA for this techniques, and then the digestion with the DNA. This is the story of DNA and how the DNA migrated towards the protein, towards the next steps. And the complex mixture of a fragment is subjected to gel electrophoresis. What is the major role of gel electrophoresis? Means that to separate the fragment according to the size. So, which is majorly it is not, don't change the size, which is majorly happened in according to the size. The restriction fragment present in the gel or denatured with alkali and transferred onto a nitrocellulose filter or nylon membrane by blotting. And the procedure was preserves in a fragment of the gel, creating in a replica of the gel filter. Is looking like it is incorporated hybridization. conditions with specific radio-labeled probe, DNA probe. The probe hybridized to the complementary DNA restriction fragment. Actually, we are thinking like what are the major steps should be there in DNA. Southern blotting means DNA should be digested and separated by the agarose gel electrophoresis, and then to detect the DNA, and by we applying the blotting techniques, and then we can do for the capillary electrophoresis techniques after we carried out from the nitrocellulose membrane. That then transfer of sodium saline citrate was can be carried out. So finally, we can detect, we can trap the targeted sequence here. Then we need to apply the complementary sequence. The sequences may be in this stage. The sequences may be present in, for example, 5' to 3'. We guess like AAT, GCAT. For the structure, we can know the complementary structure of the particular region, like we can arranging them to the perpendicular order TA, G, A, and which in a perpendicular order means 3' to 5', and complementary sequence. So this is called as complementary sequence, and prepare radio labeled sequence. Now we can prepare the radio labeled sequence. And finally, fourth step, so we applying the hybridization techniques now. What is mean by hybridization techniques? After we carry out the nitrocellulose membrane, we are having the major role for blotting, means we are carried out the sequence from the gene, and then we applied for the filter by we are using nitrocellulose membrane filter by with the action of electroboritic technique, and then to take a targeted sequence nitrocellulose from the nitrocellulose membrane. And fourth steps, we add the hybridization techniques. Hybridization techniques, now we are taking a two complementary sequence pair, like what are the two complementary sequence, like denaturation or and renaturation, and we are taking the double standard and two single standard. Detection of autoradiography charts. Detection of final step. Okay, we are applying the hybridization technique. Sequence can be paired with a probe. If the sequence can be paired with the probe, means the hybridization technique was successfully carried out. Otherwise, it is not carried in properly. By we are using the detection, final detection. How we are detecting the sequence means we are applying the technique like autoradiography by using the nitrocellulose paper, and we expose to the film with the pitch. Is the picture is visible? So emit the picture is hybridized. So done. Hybridized means hybridization done. Otherwise, the no figure can be, no picture can be, no bands can be visualized, means no hybridization can be present. So this is the major factor for the southern blotting. And this is our DNA. I will told you in before that capillary blotting. So we are taking and think like our genome. We need to take in a particular size only. So we need to cut here for the specific gene by using enzyme. Restriction enzyme to cut the particular DNA sequence, and then it will be transferred to separated DNA on agarose gel, and transfer or blot with DNA fragment from the gel to the membrane, and bound DNA bands are exposed on a film, and radio labeled probe incubated with the membrane, and the probe can be selected. If the probe can be attached with the particular sequence, means the bands can be visible. Otherwise, the bands cannot be visible. The hybridization is done, means bands can be visible. Hybridization is not done, means the band cannot be visible. So result has to be based on like that only. So membrane with the bands transfer to it. So this is the structure. And southern blotting and extraction, what is actually exactly doing? Extraction and purify DNA from. So technique is over. Extraction and purify DNA from the cell, and DNA is restricted with enzymes. Start by electrophoresis and denature DNA, transfer to nitrocellulose paper and block with excess DNA, wash off unbound probe, and autoradiography. So these are only the 8 steps to be carried out in a southern blotting. And then for northern blotting, it is also similar for southern and northern blotting. Northern blotting is a technique for detection of specific RNA sequences. Northern blotting was developed by James Alvin and George Stark at the Stanford University in 1979 and was named by analog to southern blotting. And this is rRNA. And RNA is isolated from several biological samples, for example, various tissues, various developmental stages of same tissues. Like RNA is more susceptible to degradation than DNA. And the gel is then blotted on a nylon membrane or a nitrocellulose filter paper and diazobenzylomethyl papers. Okay, diazobenzylomethyl papers. This is one of the filter, and by creating the sandwich arrangement. And samples are loaded on a gel, and the RNA samples are separated according to their size on an agarose gel. The resulting gel following after the electrophoresis run. And the membrane is placed on a dish containing the hybridization buffer with a labeled probe. Thus, it will be hybridized to the RNA on the blot that corresponds to the sequence of interest. And the membrane is washed to remove unbound probe. The labeled probe is detected via autoradiography and chemiluminescence techniques. And we know actually the probe is visible means the process can be the similar RNA can be there, otherwise similar RNA cannot be there. And a standard for the study of the gene expression at the level of mRNA, so messenger RNA transcript. So detection of mRNA transcript size and study RNA degradation and study RNA splicing and study RNA half-life and also study for after using to confirm and check transgenic knockout mice for the animals. And standard northern blot method is relatively less sensitive, and detection with multiple probes is a problem, and it is RNA samples are even slightly degraded by RNAs. The quality of DNA and quantitation of expression is quite negatively affected. Western blotting. So Western blotting means what? We need to detect some of a protein. We need to detect protein compound. At the Western blotting, it was carried out in the year of 1981. Is an immunoblotting technique which rely on the specificity of binding between the protein of interest and the probe. So antibody raised against the particular protein to allow the detection of protein of interest in a mixture of many other similar molecules. So only one difference, we are taking a gel from previously used two techniques, we are using the gel. Here we are using polyacrylamide gel electrophoresis. So that is only the difference. STS page technique is prerequisite for Western blotting. So separation, transfer, staining, and visualization. These are the 4 methods should be carried out in Western blotting. And Western blotting, a protein sample is subjected to electrophoresis and an STS page polyacrylamide gel electrophoresis. So this is only the difference in before the 2 blottings and for the Western blotting. And the electro transfer separated protein from the gel to surface of the nitrocellulose membrane. So this is the structure of Western blotting. The blot is incubated with a genetic protein such as milk proteins or BSA, which bind to any remaining sticky places on the nitrocellulose, an antibody that is specific for the protein of interest. After washing, the washing and removal of nylon specificity, and we bound AB1 and secondary antibody. Actually, this is sandwich method to add AB2 is added, which specifically recognize the domain. So primary antibody and secondary antibody that can be covalently linked to report with the enzymes, which allow the visualize the protein AB1 and AB2 complex. Conclusion. The conclusion is basis on old blotting or new sequencing techniques to better determination the role of specific biomolecules, protein, DNA, RNA, and for the clinical purposes. And finally, I will give the little introduction to the conclusion. Yeah, it is over for the blotting, but we regarding like don't forget hereafter what is mean by blotting technique. You can remember like Parvati ma'am class also. And what is mean by blotting now? We need to transfer. We need to transfer of gene or need to detect DNA, RNA and protein. So we need to plan for DNA and then we need to digest with the DNA with the help of restriction endonuclease enzymes and add the gel electrophoretic technique and then to separate the DNA and then to use mild NaOH double standard DNA to transfer to single standard DNA. This technique is known as hybridization techniques, and then we loaded it for again in a blotting and then to filter plan for filtration by using nitrocellulose membrane filter, and we track the DNA. After tracking the DNA, hybridization technique so has to be carried out in before the tracking. Okay, tracking the sequence from the nitrocellulose membrane, and then we applied for the hybridization techniques, and in that specific hybridization techniques which have in a renaturation and denaturation process, high temperature, and we apply low temperature to digest the double standard DNA to single standard DNA, and after we apply in a similar mild heat, after it can be renatured, it can be again converted to double standard DNA, and then we apply the radio labeled probe. The probe. Then after the hybridization, we use as some of the probe. That probe is can be fixed with our molecules or not, and by we checking the technique is known as radioactive technique, and then finally it can be probed with our sequence. Means the visible band can be visible. The band can be visible means our process can be success. The band can be not visible means the absence of DNA can be present. So this is enough for the blotting techniques. Thank you.
Thank you, ma'am. We really enjoyed your
Yeah.
presentation, ma'am. It was really very engaging.
Okay.
Over to Anshu. We shall welcome Deepa. Over to Anshu.
Yes, sir. Good evening, everyone. Deepa ji is a third year student in microbiology and chemistry. She is studying in University College, Mangalore, Karnataka. She has a strong interest in advancing scientific knowledge and hence attends webinars and workshops to keep up with the recent advancement. She is eager to pursue her higher studies and research in life sciences with the goal of contributing to innovative solutions in microbiology, biochemistry, and related fields. She is committed to building a career in research and applied sciences through continuous learning and practical exploration. Apart from her studies, she is interested in public speaking, reading, and writing. She is an active member of the English Association in her college and actively takes part in competitions held by the association. She has won third prize in pick and speak competition in her college and she has achieved district level victory in BITS National Spell Bee competition during her years. So I welcome Deepa ji.
Ma'am, your mic is not on.
Okay, my apologies. Thanks for the warm welcome. And before I jump into the topic, I would really like to thank everyone for this wonderful opportunity for allowing me to speak on this day and also for giving me this wonderful opportunity to speak about feminism and the mm, concepts of feminism. So, today I would like to speak about what is feminism and who is a feminist, and also about what are the problems that are faced by the feminist. So let's start. So is the screen visible?
Yes, ma'am.
Okay. So before we start, let me say that I'm not here to hate on anybody and I'm not here to offend anyone. So I'm just spreading the awareness about this topic, feminism. So without any delay, let's jump into the definition of feminism. So, what is feminism? Feminism, in simple words, means fighting for equal rights. It asks society to give women the same respect, chances, and opportunities as men. Feminism reminds us that women and men are equally capable, which means that talent, intelligence, and strength are not decided by gender. Feminism also breaks stereotypes. It says that a woman can choose her own path just like a man and in all aspects. So, feminism is about fairness and not superiority. So feminism doesn't put women above men, it makes sure that both stand on the same ground. This is really an important factor because this is misunderstood most of times because whenever we talk about feminism, people see it as a superiority. Actually, it's not. We are just talking about fairness in here. So let's move on to next point, which is feminism gives women a voice. It ensures that women's opinions, choices, and dreams are heard and valued in every space. So it just gives equal opportunities and it lets a woman to choose for herself, and it also lets a woman to dream what she wants in every field. It is not condemned to one particular field or in one space. So it's uh, applicable in all the fields. So the next point is feminism builds a bright society, a society where respects and opportunities are shared equally, creating harmony between men and women. And this is why feminism is really needed because the society nowadays don't show equality for both women and men. So feminism makes sure that a bright society is built and harmony is built between men and women. So let's move on to the question, who is a feminist? In simple words, we can say that a feminist is anyone who believes in feminism. A feminist is someone who stands against discrimination and supports fairness for all genders. A feminist need not be only women. It can also be a man as long as they believe in equality and support equality. So again, a feminist is usually seen as a woman, and it need not be so. A man can also be a feminist as long as he believes in the true meaning of feminism. So let's move on to the question, why is feminism needed? we have already discussed the definition of feminism. So let's see why is feminism needed. In the history, feminism has brought many victories to this world. So let's move. So let's see what are those victories and um, how was it achieved. So first thing is right to vote movement. It's also known as suffrage movement. This was led by women like Susan B. Anthony and Elizabeth Cady Stanton in the US and Emmeline Pankhurst in the UK. They fought for decades until women finally gained the right to vote. So before we move forward, we shall look into who is Elizabeth Cady Stanton and also we will look into other characters. So Susan B. Anthony, she lived from 1820 to 1906, was one of the most important leaders of the women's rights movement in America. Born into a Quaker family that believed in equality, she grew up with strong values of justice and fairness. She began her career as a teacher but soon realized that women had very few rights compared to men, both in education and in society. This inspired her to dedicate her life to fighting for equal rights, especially the right for women to vote. Alongside Elizabeth Cady Stanton, she co founded the National Women's Suffrage Association in 1869. Together, they gave speeches, wrote articles, and organized campaigns demanding voting rights for women. Susan was bold and fearless. She even cast a vote in the 1872 US election when it was illegal for women to do so. For this act, she was arrested, but instead of being silent, she used her trial to bring national attention to the cause of women's suffrage. Although Susan B. Anthony did not live to see women win the vote, her efforts laid the foundation for change. 14 years after her death, in 1920, the 19th Amendment was passed, granting women the right to vote in the United States. This amendment is often called the Susan B. Anthony Amendment in her honor. She remains an enduring symbol of courage, determination, and the belief that women and men deserve equal voices in shaping society. So, uh, let's move on to the next point, which is known as education for girls. In India, Savitri Bai Phule was the first female teacher who opened schools for girls, and globally, Malala Yousafzai became a symbol of modern feminism by risking her life to demand education for every girl. So, before we move on, we shall see who uh, Emmeline Pankhurst was. Ali, before we move on, we shall see who was Elizabeth Cady Stanton because she came next to the Anthony Elizabeth, Susan B. Anthony. So, Elizabeth Cady Stanton was a bold and visionary leader of the women's rights movement in the United States. Born into a well off family in New York, she received more education than most women of her time, which gave her the confidence to change inequality. She noticed early on that women were denied the same legal rights as men, especially in areas like property ownership, education, and divorce. This deep sense of injustice fueled her lifelong mission to demand equality for women in every part of the society. She made history by organizing the first women's rights convention at Seneca Falls in 1848. At this convention, she presented the famous Declaration of Sentiments, a groundbreaking document that demanded equal rights for women, including the right to vote. Her words challenged society's deeply rooted traditions and shook the foundations of the legal system that kept women dependent on men. Stanton also worked with Susan B. Anthony for over 50 years, writing speeches and petitions and books that pushed the movement forward. While Anthony often delivered speeches in public, Stanton provided the powerful ideas, arguments, and writings that gave the suffrage movement its intellectual strength. Um, Beyond voting rights, Stanton also spoke about issues that were considered radical in her time. She advocated for women's control over their own bodies, equal opportunities in education and employment, and reforms in marriage and diverse laws to protect women's independence. Even though she did not live long enough to see the passage of the 19th Amendment in 1920, her vision paved the way for it. Today, Elizabeth Cady Stanton is remembered not only as a suffragist but also a thinker, writer, and reformer whose courage expanded the very definition of women's freedom. So let's move on to about the information about Emmeline Pankhurst. So Emmeline Pankhurst lived from 1858 to 1928, and she was a revolutionary leader who changed the face of women's rights in Britain. At a time when women had no say in politics, she believed that peaceful speeches and polite petitions were not enough. She argued that only bold, dramatic action could wake up society to the injustice women were facing. To fight this battle, she founded the Women's Social and Political Union, which was also known as WSPU in 1903, which quickly became famous for its fearless methods. Members of her groups called suffragettes changed themselves to railings, broke windows, staged hunger strikes, and interrupted political meetings. These actions shocked the nation and even landed Emmeline in jail many times, but every arrest only made her stronger and spread the message further. Her struggle finally bore fruit when women in Britain won the right to vote in 1918. Though the law at first applied only to certain women over 30, it was the first step, which was the big step for the equality. 10 years later, Britain granted women equal voting rights with men, a dream that Emmeline had fought for her whole life. Today, she is remembered not just as a suffragette, but as a fearless fighter who believed that deeds, not words, could change the world. So let's now move on to the information about Savitri Bai Phule. She lived from 1831 to 1897, and she was one of the most remarkable social reformers in India. Remembered as the country's first female teacher. Born in Naigaon, Maharashtra, she faced intense discrimination because of her caste and gender. Yet, with the support of her husband, Jyotirao Phule, she defied all the odds and pursued education at a time when it was considered unacceptable for women, especially from marginalized communities, to learn. Her courage marked the beginning of a revolution in women's education in India. In 1848, Savitri Bai and Jyoti Rao established the first school for girls in Pune. This act was revolutionary and angered conservative sections of society who often harassed her by throwing mud, cow dung, and stones as she walked to school. Despite such hostility, she continued teaching, often carrying a space sari to change into when people tried to shame her. Her perseverance and sent a powerful message that education was not a privilege reserved for men or higher caste, but a right for every human being. Beyond education, Savitri Bai was a passionate advocate for social justice. She and Jyotirao worked tirelessly against caste-based oppression and the passion of untouchability. Together, they set up the Satyashodak Samaj, which was also known as Truth Seeker Society, which challenged caste hierarchies and worked towards equality. She also opened shelters for widows and orphans, providing them with a dignified life when society rejected them. Savitri Bai Phule also raised her voice against child marriage and supported widow remarriage, which were taboo during her time. She often wrote poetry that carried themes of empowerment, self respect, and the importance of education. Her writings inspired countless women to believe in their own strength and capabilities, planting seeds of feminism in Indian soil long before the term became popular. Her legacy remains timeless. Today, Savitri Bai Phule is remembered not just as a pioneer of women's education, but as a fearless reformer who fought for equality on multiple fronts: gender, caste, and class. She transformed the lives of countless marginalized individuals and paved the way for future movements for women's rights in India. Her story stands as a reminder that true change begins with courage, compassion, and determination to challenge injustice. So, uh, we will also look at one of the part of her poem. She said that go get education. be self reliant, be industrious, work, gather wisdom and riches. All gets lost without knowledge. We became animals without wisdom. Sit idle no more. Go get education. End misery of the oppressed and forsaken. You have got a golden chance to learn. So learn and break the chains of caste. Throw away the veil and go get education. Be self reliant, be industrious, work, gather wisdom and riches. So this was about Savitribai Phule. Now let's move on to the next point, which is workplace equality. So Gloria Steinem was a strong voice in the US feminist movement, fighting for equal pay and opportunities. In India, Dr. B.R. Ambedkar worked to include women's rights in the constitution, ensuring workplace dignity and legal protection. So am I audible?
Yes, ma'am.
Yes.
Okay, and do you guys see the screen?
Yes, ma'am.
Okay. So uh, let's uh, let's see about the information about Gloria Steinem. So Gloria Steinem was born in 1934 and she was an American feminist, journalist, and social political activist who became one of the most recognized faces of the women's liberation movement in the 20th century. Born in Ohio, she experienced early challenges with family instability, which made her determined to fight for equality. Her education and career in journalism became the tools through which she raised awareness about women's rights. In the late 1960s, Steinem co founded Miss Magazine, a groundbreaking publication that focused on feminist issues, women's rights, and social justice. Through her writing, she addressed topics that were largely ignored by mainstream media, including workplace discrimination, reproductive rights, and domestic violence. Her work helped bring feminist ideas into American homes and sparked widespread discussion on gender equality. Steinem was a vocal leader in campaigns for the Equal Rights Amendment, also known as ERA, pushing for illegal equality for women in the United States. She organized rallies, gave speeches, and collaborated with other activists to ensure women had a voice in shaping laws and policies. She also connected feminism with broader social justice issues, including racial equality and labor rights. Beyond activism, Gloria Steinem became a symbol of the feminist movement globally. She used media appearances, books, and public speaking to inspire women to challenge societal norms and embrace their rights. Her commitment to nonviolence, education, and advocacy helped to create a culture where women could openly demand equality. Today, Steinem's legacy continues to influence generation of activists. Her work paved the way for modern feminist movements, showing that change is possible through education, dialogue, and persistence. She remains a beacon for those fighting for gender equality, reminding the world that feminism is about fairness, opportunity, and respect for all. So, in workplace equality point, we have also discussed about Dr. B.R. Ambedkar. So let's also see the information of Dr. B.R. Ambedkar. So Dr. B.R. Ambedkar lived from 1891 to 1956 and he was an Indian jurist, economist, social reformer and the principal architect of Indian constitution. He was born into a Dalit family. He faced severe caste discrimination and social exclusion from an early stage. These experiences shaped his lifelong mission to fight for equality, justice, and the rights of marginalized communities, including women. Ambedkar strongly believed that social reform and legal equality were essential for progress. He campaigned against untouchability, caste-based discrimination, but he also emphasized women's rights as a critical part of social justice. He argued that gender equality and caste equality must go hand in hand for a fair society. One of his major contributions to women's rights was drafting progressive laws in the Indian Constitution. He ensured provisions for equality before the law, the abolition of discrimination, and the protection of women's property and inheritance rights. He also supported the Hindu Code Bill, which sought to give women rights in marriage, inheritance, and divorce. radical reforms for that area. Ambedkar's advocacy extended beyond legislation. He encouraged education for women and marginalized communities, believing that knowledge was a powerful tool to fight oppression. He worked tirelessly to create awareness about women's legal and social rights, showing that true empowerment requires both law and education. Today, Dr. B.R. Ambedkar is remembered as a champion of equality and justice. His work transformed Indian society by giving legal backing to the fight against caste and gender discrimination. He remains an inspiration for feminists and social reformers, demonstrating that structural change combined with social awareness can create a fair and just society for all. So, coming to the next point, it's about legal rights and social reforms. Rajaram Mohan Roy campaigned against practices like sati and child marriages, paving the way for legal reforms. Later, Begum Roke fought for women's rights in education and social life. So let's move on to the information about Raja Ram Mohan Roy. So Raja Ram Mohan Roy was a prominent Indian social reformer, scholar, and thinker, often called the father of modern India. Born in Radhanagar, Bengal, he was well educated in Sanskrit, Persian, Arabic, and English. This multilingual education helped him understand both Indian traditions and Western thought, giving him a unique perspective on society and reform. He strongly opposed social evils like sati, child marriage, and caste discrimination, witnessing the practice of sati, where widows were forced to immolate themselves on their husband's funeral pyres, deeply disturbed him. He campaigned tirelessly against this practice, arguing that it was against both morality and true religious principles. His efforts eventually led to the Bengal Sati Regulation of 1829, which legally abolished sati in British India. Rajaram Mohan Roy was a pioneer in promoting women's rights and education. He believed that education was the key to social reform and urged the establishment of schools for girls. He worked to ensure that women could learn reading, writing, and arithmetic as well as understand their rights in society. His advocacy laid the groundwork for later reformers like Savitribai Phule to advance women's education further. He founded the Brahmo Samaj in 1828, a socio-religious reform movement aimed at purifying Hinduism and promoting monotheism. Through the Brahma Samaj, he encouraged the rational thinking, opposed idolatry, and promoted social equality. The organization became a platform for many other reformers to challenge orthodox practices and push for modernization in society, such as Indian society. So, Rajaram Mohan Roy also played a key role in political and economic reform. He petitioned the British government for free press, legal reforms, and the protection of Indian interest. He supported freedom of expression, and his writings often advocated for administrative improvements, land reforms, and economic policies to reduce exploitation of peasants. He was a scholar, writer, and linguist, fluent in several languages, and used his knowledge to bridge Indian and Western thought. He wrote intensively on religion, social issues, and philosophy, emphasizing the reason and human rights. His work helped Indians understand reform not as a rejection of culture but as an evolution toward justice, equality, and enlightenment. Rajaram Mohan Roy's legacy is immense and enduring. He is remembered as a visionary who combined knowledge, courage, and moral conviction to challenge social justice by fighting against oppressive practices, promoting women's education, and encouraging rational thinking. He laid the foundation of modern Indian society. His ideas influenced generation of reformers, making him a central figure in India's social, religious, and political awakening. So now let's move on to Begum. She lived from 1880 to 1932, and she was a pioneering social reformer, educationist, and writer from Bengal who dedicated her life to promoting education and empowerment for Muslim women. Born in a conservative Muslim family, she faced social restrictions that limited women's education, but she defied these norms and pursued knowledge. Her early experiences motivated her to work for the upliftment of the women in society. She strongly believed that education was key to women's empowerment. In 1911, she established the Sakhavat Memorial Girls School in Kolkata, the first school for Muslim girls in the region. Despite facing strong opposition from conservative sections of the society, she preserved, insisting that every girl, regardless of her background, deserved the right to learn. Begum Rokeya was also a powerful writer and thinker. Her book Sultana's Dream, a feminist utopian novel, imagined a world where women led society and men were confined to domestic roles. And the work challenged patriarchy, promoted gender equality, and became an inspiration for feminist thinkers in India and beyond. She worked tirelessly to reform social practices that oppressed women. Begum Ruqayya campaigned against child marriage, purda, and other restrictions that limited to women's freedom. She also encouraged women to participate in public life, learn skills, and become self reliant. Her work laid the foundation of modern feminist movements among Muslim women in India and Bangladesh. Her legacy continues to inspire generations. Begum Ruqayya's vision of an educated, independent, and respected woman influenced countless reformers and educators. Institutions like the Begum University in Bangladesh stand as a tribute to her work. Today, she is remembered as a fearless advocate for women's rights whose encouragement and intellect challenged societal norms and transformed the lives of women in South Asia. So now let's move on to a next point, which says that feminism helped to fight against dowry and domestic violence. So Ishwar Chandra Vidyasagar in India worked to pass the Widow Remarriage Act and campaigned against social oppression against women. So let's know about Ishwar Chandra Vidyasagar. He lived from 1820 to 1891 and he was a towering scholar, educator and social reformer of 19th century India. Born in Bengal into a poor Brahmin family, he faced many hardships during his childhood. Despite financial difficulties, he excelled in studies, earning the title Vidyasagar, which means that ocean of knowledge. For his extraordinary command over Sanskrit, Bengali, and English, his dedication to learning became the foundation for his lifelong mission of social reform. Education was the cornerstone of Vidyasagar's vision for society. He believed that country could only progress if women and marginalized communities were educated. To this end, he established and promoted numerous schools for girls in Bengal, challenging the deeply entrenched belief that women did not need a formal education. He also encouraged modern education among boys, emphasizing rational thinking and knowledge over blind tradition. One of his most notable contributions was his advocacy for widow marriage. In 19th century India, widows were subjected to severe social restrictions, often condemned to a life of deprivation and isolation. Vidyasagar campaigned relentlessly for the Hindus' Widow Remarriage Act of 1856, which legalized the remarriage for widows. His efforts faced immense opposition from constitutive elements, but his unwavering commitment led to a historic change that restored dignity and hope to countless women. Vidyasagar was also a reformer of the Bengali language and literature. He simplified Bengali prose, modernized the alphabet, and authored textbooks that made learning accessible to children. His literary works were not just educational but also carried messages of morality, social justice, and empowerment. By making education understandable and practical, he ensured that learning could reach even the poorest sections of society. He was a strong advocate against social evils and blind traditions. Vidyasagar challenged the caste discrimination, excessive rituals, and other practices that oppressed women and the underprivileged. He believed that reform should come through education and rational thought rather than mere imitation of Western ideas. His approach combined respect for Indian culture with a progressive vision for social justice. Beyond this educational and social work, Vidyasagar was deeply involved in policy and institutional reforms. He served as a member of the Education Department of Bengal, advancing on curricular reforms, teacher training, and the expansion of schools. He worked tirelessly to ensure that reforms were practical, sustainable, and had a real impact on society rather than being just symbolic gestures. His life was a model of simplicity, integrity, and dedication. Despite his fame and influence, Vidyasagar lived modestly, focusing on the welfare of society rather than personal gain. His efforts transformed the lives of women and children, laying the foundation for a more enlightened and equitable society in India. Ishwar Chandra Vidyasagar's legacy continues to inspire generations. He is remembered not only as an educator and scholar, but as a fearless reformer who challenged entrenched social norms to promote equality, justice, and empowerment. His work in women's education and widow remarriage remains a beacon of progressive thought, making him one of the most influential figures of Indian social reform history. So now let's move on to the next point, reproduction rights and health. So Margaret Sanger in the US campaigned for birth control access and women's reproductive freedom. So let's know about Margaret Sanger. She lived from 1879 to 1966. And she was an American activist, nurse, and pioneering advocate for women's reproductive rights. Born in Corning, New York, she worked witnessing her mother struggle with multiple. Sorry for the delay. She was born in Corning, New York. She grew up witnessing her mother struggle with multiple pregnancy and poor health, which left a lasting impression on her. From an early age, she became determined to fight for women's access to birth control and the right to make choices about their own bodies. Sanger began her work as a nurse, where she saw the devastating effects of uncontrolled pregnancies on women's health. Many women died or suffered severe health problems because they lacked access to contraception and reproductive education. Her first-hand experience motivated her to educate women about family planning and to advocate her accessible birth control methods. In 1916, she opened the first birth control clinic in the United States, providing women with information and resources to prevent unwanted pregnancies. This bold step led to her arrest under the Comstock laws, which prohibited the distribution of contraceptive information. Undeterred, Sanger continued her activism, arguing that birth control was essential for women's health, freedom, and equality. Margaret Sanger also founded organizations that would evolve to Planned Parenthood, a network dedicated to reproductive health, education, and family planning. She promoted research into safe and effective contraceptives and worked to make them widely available. Her efforts not only transformed women's health but also empowered women to have control over their own lives and futures. Her legacy is both influential and controversial. Margaret Sanger is celebrated for her role in advancing reproductive rights and women's autonomy, inspiring generations of activists. While some of her views on population control have been debated, her contributions to women's health, access to birth control, and the broader feminist movement remain monumental. She is remembered as a fearless advocate for women's right to make decisions about their own bodies. So now let's move on to the next point, which is property rights. So Dr. Bina Agarwal in India highlighted women's land and property rights, which helped to empower rural women economically. So let's know about Dr. Bina Agarwal. She was born on 1951 and is a distinguished Indian economist, scholar, and feminist, widely recognized for her work on gender development and property rights. She has made pioneering contributions to understanding how social, legal, and economic structures affect women's lives, particularly in rural and other contexts. Her work combines rigorous research with a strong commitment to social justice. Born in India, Agarwal pursued her education in economics at prestigious institutions, eventually earning a PhD that focused on development issues and women's empowerment. Her academic journey allowed her to study both theoretical frameworks and ground level realities, equipping her to bridge the gap between scholarship and policymaking. One of her central areas of research is women and land rights. In her landmark book, A Field of One's Own, Agarwal demonstrates how property ownership can transform women's economic, social, and political status. She argues that secure land rights give women bargaining power within households, reduce their vulnerability to domestic violence, and improve children's well being. Agarwal's work extends beyond research to influencing policy reforms. She has advised governments, international organizations, and development agencies on gender sensitive policies. Her recommendations have informed programs that promote women's access to land, credit, and agricultural resources, ensuring that development initiatives are inclusive and equitable. She has also been instrumental in connecting theory with the grassroots activism. Agarwal collaborates with women's organizations, helping rural women claim land rights, access resources, and participate in local governance. By translating research into action, she has empowered women to assert their rights and take part in decision making at community levels. Dr. Agarwal has written extensively on the intersection of gender, economics, and social norms. Her research emphasizes that development cannot be sustainable unless women have equal access to resources and opportunities. She challenges the patriarchal practices and highlights how cultural, legal, and economic barriers often work together to marginalize women. Her contributions have been recognized globally through awards, fellowships, and positions at prestigious institutions. Agarwal's work continues to inspire academics, policymakers, and activists worldwide. Her insights into property rights, gender equity, and empowerment remain fundamental in feminist economics and development studies. Today, Dr. Beena Agarwal is celebrated not just as a scholar but as a change maker. Her work demonstrates that knowledge, when combined with activism, can transform life. By advocating for women's rights to land and resources, she has helped create more equitable societies and inspire generations to continue the struggle for gender justice. So these are some of the examples how feminists uh, help to bring freedom to India and to women. So let's move on to the next topic, like the conclusion for all this matter. So we can say from all these discussions that we can finally say feminism, most of the women from across the world gained freedom and equal rights, which made the life of women even more easier. Now, when we see women going out for education, jobs, or even to parks or theaters freely, we should remember that this freedom exists from all the sufferings and movements done by feminists. So the next question arises is that is feminism still needed? Yes, they still need even though women got most of the freedom when compared to the past generation. The reasons why feminism is needed are first point is sexual harassment. This is one of the most major problem that we face in workplaces and other public places. And we all know that increasing cases of rapes, which is extreme form of sexual harassment. So feminists are raising voices against it, which is very important. So the next point is stereotypes and social pressure. Women are still judged for their choices in career and marriage, appearance and lifestyle, limiting freedom and opportunities. Best example is women are judged for wearing sleeveless dresses, shorts, etc. But men are not judged for whatever they wear or whatever they say about women. So the next point is reproductive rights. So many women do not have full control over their own bodies, healthcare, and family planning decisions. Best example is that in most of the hospitals, they ask for signatures of husbands to do the family planning procedures, which is very common in most of the hospitals. So moving on to next point, which is job opportunities. This is one of the most important problem that must be noticed very early, because in most of the STEM fields, job applications specifically mention that men are preferred, which decreases the employment opportunities for women, and women are forced to take other than STEM fields, even though women are interested in those fields. So now we discussed what is feminism, who are feminists, and we also seen what are the advantages of feminism. So now let's discuss what is toxic feminism. Toxic feminism is when the idea of equality is misused to blame or hate men instead of promoting fairness and understanding between genders. It focuses on superiority rather than equality, turning feminism into anger or division rather than a movement for everyone's rights. So this is toxic feminism, which is often uh, confused with the real meaning of feminism. So we now know what toxic feminism truly is and what truly the feminism means. So moving on to the problem of feminist. So as discussed earlier, toxic feminism made social media to hate feminism. This made people to hate feminism and they even hate the feminist. This is the major problem because most of the feminists are getting hate nowadays because of toxic feminism. Nowadays, the feminist can't say that they are feminist and they can't just introduce themselves because of the hate and the weird clicks. So, from all these discussions, I want to say that the ultimate message of this presentation is to say that never mistake feminism for toxic feminism because even I have faced such scenarios where I was judged for saying that I'm a feminist. Even in my college, feminists are made fun even by the professors. Professors say that women should leave feminism because it's toxic and they just say things which are not real because it's actually really anger. It's literally very sad that we see feminism as such a negativity factor because feminism, because of them, we our sisters, our mothers, has freedom now, and because of the feminist, we are able to see women freely in smiling figures and also very happy. So let's respect feminism. I'm not saying everyone to become a feminist or to follow feminism. I just want everyone to respect feminism and also the feminist. So with this, I want to end my discussion. Thank you everyone for your patience.
Thank you so much, Deepa ma'am. You explained everything so well. It was really pleasure listening. Okay, now Anshu, you can introduce Shravya ma'am.
Yes, ma'am. Bojala Shravya is an emerging researcher with a diverse background in biotechnology, microbiology, and nanoscience. Her experience spans academic research and industry exposure, reflecting a commitment to innovation, scientific rigor, and societal impact. Shravya's expertise lies in R&D at the interface of nanotechnology and life sciences, with emphasis on nanoparticle synthesis and characterization, phytochemical analysis, and cell structure. Techniques oriented towards biomedical and environmental applications. She has presented her research at national and international conferences, earning recognition for her ability to communicate complex scientific concepts with clarity and impact. Alongside conference participation, she has authored and co-authored peer-reviewed scientific publications showcasing her ability to contribute to the growing body of global research. Beyond technical expertise, she is passionate about the intersection of nanotechnology, medicine, and sustainability. Her research interests are guided by a vision of translating laboratory advancements into practical solutions for human health and environmental well-being. This combination of technical depth, research, acumen, and forward-thinking perspective positions her as a promising contributor to the future of science and technology. So I welcome Ms. Shravya.
Thank you, Anshu. Yeah. Good evening, everyone. As it was late, I'll try to finish as early as possible. So, I'm delighted to be here with you today. So, our topic is microbial techniques. That means the tools that let us study and work with microorganisms. Now, when we think of microorganisms or microbes, the first thought is usually a disease causing germs, right? But microbes are far more than that. They are the foundation of ecosystems, key to human health, and partners in food, energy, and also industry. Let me give a perspective. Microbes existed billions of years before humans, right? They shape the Earth's atmosphere, they draw the nitrogen cycle that plants depend on, and they still live inside us in numbers greater than our own cells. In fact, without microbes, there would be no yogurt, no bread, no antibiotics, and even no vaccines, also. So, how do we understand them? Just as an astronomer needs telescope to study stars, like that, a microbiologist need techniques. That means culture, microscopy, staining, molecular methods, and some biochemical tests also. So these are not just lab tricks, they are the reason we can detect diseases quickly, develop medicines, and harness microbes for industry and the environment also. Coming to the culture techniques, it was one of the oldest and most essential tools is microbial culture. Imagine if you had a room full of people all talking at once. To study one person, you would need them to speak individually. Similarly, microbes also exist in mixed communities. To study one species, we need pure cultures. So that we are going to divide into three types of techniques. That is nothing but streak plate, pour plate, and spread plate. So, what is this streak plate? If we drag a loop of bacteria across agar plates so that individual cells get separated and grow into distinct colonies. Actually, this method was a key in Robert Koch's work, proving that anthrax and tuberculosis were caused by single microbes. The next is pour plate method. In this, microbes are mixed into melted agar and colonies develop inside the medium. This is useful for the estimating microbial counts in water or milk. Next is spread plate method. In this, samples are spread evenly over agar, commonly used in select if in case of selective media, it means example McConkey agar selects gram negatives. If in case of differential media, help us to distinguish microbes, example blood agar shows hemolysis patterns. So these techniques may look simple, but they revolutionized medicine without pure cultures. So Koch postulates that means the foundation of germ theory would never have been established. Next, coming to the microscopic techniques. If once microbes are cultured, we want to see them, so that microscopy is the window into this hidden world. Let me say that there are two types of microscopy. One is light microscopy and other is electron microscopy. Coming to the light microscopy, it was a bright field microscope. That means classrooms are standard, often requiring staining. Phase contrast and differential interference contrast allow us to see living unstained cells in motion, watching protozoa swim or bacteria division also. Coming to the electron microscopy, when scientists wanted to see viruses for the first time, they needed these electron microscopes. And this electron microscope is divided into SEM and TEM. What is the difference between these SEM and TEM? Let me say, SEM means scanning electron microscope. That means it gives detailed 3D images. Imagine seeing the hair-like play of bacteria or the spiky coat of coronaviruses. Coming to the TEM is a transmission electron microscope. It goes deeper, showing internal structures like ribosomes, membranes, viral particles inside the host cells also. The last is fluorescent microscope. That means by tagging proteins with fluorescent dyes or GFP, that means green fluorescent protein, scientists can track process inside living microbes. For example, seeing how bacteria form biofilms or how antibiotics disrupt cell division. Every breakthrough in microbiology, from discovering E. coli pili to understanding the COVID-19, has relied on microscope only. Next, coming to staining methods. This staining makes microbes visible and differentiates them. So, in this, first we'll talk about gram staining. So, this gram staining was first developed in year 1884 by Hans Christian Gram. Hence, it was named as gram staining. It is a simple test. It guides doctors today. Suppose a gram positive bacteria appeared purple, gram negatives in pink. Why is this important? It's a general question. Because gram negatives with their double membranes are often more drug resistant. Next is acid fast staining. It is a key for identifying Mycobacterium tuberculosis. Without it, early TB detection would be far more difficult. I think you got it. Next is special stains. In these special stains, we are more focusing on spore staining, capsule staining, and flagella staining. First one is spore staining. It reveals dormant but highly resistant spores of Bacillus and Clostridium. Coming to capsule staining, it highlights protective capsules which helps bacteria evade immune systems. Next is flagella staining. It shows the tiny whip-like structures used for motility. These all staining methods are not just academic, they guide diagnosis, treatment, and understanding how pathogens survive. Next is molecular techniques. Now let's fast forward to modern era. Culture and staining are powerful, but some microbes can't be grown in labs at all. Enter molecular biology. That means PCR, that means polymerase chain reaction. It's like a photocopier of just like DNA. With just a tiny sample, we can amplify millions of copies. So, for example, during the COVID-19 pandemic, PCR became a household word. I think all of you know that. It let us detect the virus quickly and accurately also. So, like that, next is DNA sequencing and metagenomics. Instead of culturing, as I said above, we can directly sequence DNA from soil, oceans, or the human gut also. This has revealed the vast microbial dark matter, organisms we have never cultured but know only by their genes only. Next is CRISPR. It's originally discovered as a bacterial immune system against viruses. This CRISPR is now a powerful gene editing tool. In coming to the microbiology, it lets us design microbes that produce medicines, break down pollutants, or detect pathogens in minutes. These molecular tools have transformed microbiology into a data rich, and we can also say that it was a high tech in science. Next is biochemical techniques. Next, you may ask, what can microbes do? The first thing is enzyme and metabolic test. It means catalase test. What does catalase test do? It detects oxygen bubbles from hydrogen peroxide, that is H2O2. And also urease test. What is this urease test? It is important for Helicobacter pylori detection. And last is carbohydrate fermentation tests. These are bread and butter in every clinical labs. Next is antibiotic susceptibility testing. That means it was famous by Kirby Bohr disk diffusion test. where antibiotics diffuse into agar and showing clear zones if bacteria are sensitive. These tests literally saves lives every day by guiding therapy. Next is fermentation studies and biochemical tests. So, from bread and wine in ancient times to antibiotics, biofuels, and probiotics today also we are using. Fermentation is how humans have partnered with microbes for centuries. So biochemical techniques bridge laboratory science with real world applications also. Next, coming to the applications, why do these techniques matter? Let's look at some applications. First is medical microbiology. That means diagnostics, vaccines. Diagnosing infections, designing vaccines, and even developing antibiotics all depend on these techniques. For example, PCR and sequencing identified SARS CoV 2 within weeks of its emergence. Next is industrial microbiology, probiotics, and fermentation. Microbes produce antibiotics, right? And also they produce vitamins, enzymes, probiotics, and even bioethanol fuel also. The probiotic industry alone is worth billions of dollars today. The next is environmental microbiology. So, as we say the environmental first, we will be reminding of wastewater treatment. Either it may be solid waste, or else liquid waste, oil spill cleanup, and plastic degradation. Microbes are nature's recyclers. One famous example is Pseudomonas bacteria. It is used to clean up spoil spills by breaking down hydrocarbons. Everyone we look medicine, food, environment, industry, microbes are silent workers. The last but not least is challenges and future. It's not all smooth sailing. Contamination is always a risk. Even experienced labs also struggle with keeping cultures pure. Antibiotic resistance is one of the greatest threats, microbes evolving into superbugs. And coming to the cost of advanced equipment, it limits research in developing countries. But the future is bright when automation and robotics are streamlining labs, and even nowadays AI is a trending, that is artificial intelligence. It helps to analyze massive sequencing data sets. And next is synthetic biology. It allows to design microbes that capture carbon, produce clean energy, or even generate new drugs. And portable sequences now allow microbial studies in the field from rainforests to space stations. We are entering an age where microbes may help solve some of the humanity's biggest problems like climate change, food security, and even health crises also. Coming to the conclusion, to conclude, these microbial techniques are the foundation of microbiology from Robert Koch agar plate to CRISPR gene editing. Each method adds a layer to our understanding. Together, they have given us life saving medicines, sustainable industries, and insights into our hidden microbial world also. The story of these microbes is not just about their disease, it's about a partnership, innovation, and even survival. As we continue to match traditional methods with cutting edge technology, microbial research will remain central to global progress in health, environment, and biotechnology. Thank you very much for your attention. I would be glad to be here with you. Thank you.
Thank you, ma'am. Thank you so much. That was an excellent presentation, ma'am.
Thank you.
Okay, now, Anagha, ma'am, can you hear me? Anagha, ma'am, are you here? Okay, Parvati, ma'am, are you here? Deepa, ma'am, can you hear me?
Yes, ma'am, I can hear you.
Okay, Deepa ma'am, I have a question for you. Can you please turn on your camera?
Okay, ma'am, just a second.
Yes, ma'am. I
Yeah.
have a question for you. What is the role of patriarchy in perpetuating gender inequality?
Can you please repeat the question?
Yes, ma'am. What is the role of patriarchy in perpetuating gender inequality?
Incorporating?
Perpetuating gender inequality. I'll just repeat it once again,
The
ma'am.
voice is very
Okay,
okay.
I'll just repeat it. What is the role of patriarchy in perpetuating gender inequality?
Mm, I'm not able to hear that one sentence in between or the incorporating.
No, no, in perpetuating.
Okay.
Ma'am, your mic is not on, I think. You're not audible.
I'll just rejoin again. Wait a second. I have some
Ma'am,
issues.
your voice is very low. Ma'am, I've dropped the question in the chat box. Yes, Deepa ma'am, we are waiting for your answer.
Ma'am, am I able to? Sir, is it-
Ma'am, your voice is cracking. Yes, ma'am.
Okay, can you please rephrase the question?
Yes, ma'am. What is the role of patriarchy in perpetuating gender inequality? I have dropped the question in the chat box, ma'am. You can see if I am not audible to you.
Okay, cause yeah, there are some blur in the voice, so I will just look at the question.
Okay, ma'am.
Sorry, I'm having network issues. I'll be answering your question, but if my profile gets exited again, I'm really sorry.
Okay, ma'am.
Ma'am, can you please put the question in the chat box?
Yes, ma'am, I've already put. Please check, ma'am.
I have checked it, but maybe because of rejoining, I can't see it.
Just a minute, ma'am. I'm sending it to you again. Yes, ma'am, I have resent it. Okay, I think Deepa ma'am has some network issue her side. So, Shravya ma'am, are you here? Shravya ma'am, are you here? Can you hear me? Sarath, I think all the four speakers they have left the meeting or they have some network issue. So I think I should proceed with the vote of thanks. Okay, so now I shall be proceeding with vote of thanks. Just a minute. Ladies and gentlemen, esteemed guests, speakers, and organizers, I, Varisha Faruqi, Secretary of the Organizing Committee from Uttar Pradesh, would like to extend my heartfelt gratitude to everyone who contributed to the success of this wonderful online talk session. Heartiest thanks to our guest speakers, Dr. K. Parvati from Tamil Nadu, Ms. PG Anaga from Kerala, Ms. Deepa Ji from Karnataka, and Ms. B. Shravya from Andhra Pradesh for sharing their valuable insights and expertise to students and fellow participants in this program. Really, your lecture and the presentations were truly inspiring and successfully catered the expectations of everyone present over here today. Thank you so much for your informative lecture. We appreciate the time and effort taken by all participants to engage in meaningful discussions and share their perspectives. Your contributions have enriched the event experience. A special thanks to Mr. Sarath, the director of Nosrex Audentia's OPC Private Limited, for his tireless efforts in planning and executing this program, and also to our honorable chairman of the Academic Events Management Committee, Dr. Kailash Chandra Srivastav sir, and Professor Dr. Rashmi Mishra ma'am, the honorary secretary, for their gracious presence and invaluable support. As we conclude today's session, we take away valuable knowledge and insights that will inspire us to make a positive impact. Thank you all for being part of this enriching experience. Thank you once again to everyone involved, and I wish you all a wonderful day ahead. Thank you, everyone. Okay, so now everyone can leave.
Thank you.